What we've done today, we're looking at manufacturing scale-up and manufacturing scale-up in a little different way than I think has been looked at before, at least on a large scale in this industry. There's been there's been challenges with the industry, as we know, and we're all very good at talking about those challenges and identifying all of the hurdles that are between us and having deliverable therapeutics. But some of what's happened over the course of time, at least in our experience after plowing away at this for a long time, is that the solutions are beginning to form very neatly around the unique challenges that are presented to the industry. And we keep hearing that we are the biologics industry of 20 years ago. At one point we heard we were the biologics industry of 5 years ago, then it was 10 years ago, now it's 20 years ago. And so we start thinking, what is it we can learn from the biologics industry? And clearly the therapies from the biologics industry, which at one time were just science experiments, as I might say many of our current therapies are, somehow they became deliverable. Somehow the people that worked in that industry and on those products figured out a way to make them deliverable. So how do we form deliverable? One of the premises I like to start with is talking about the product being the process and the process being the product. And I think we all understand what that means. But every time I hear it, I get a little bit concerned. Because what it says is if the process is the product, then my contention is you don't have a product, particularly with the manufacturing processes the way they are today. Manufacturing processes being immature, they need an awful lot of work, and they do actually challenge us in business model and in deliverability. So how do we determine deliverability? We'd like to say, yes, we understand that's true. Process is product, that's true. But it's not the entire story. The product is much, much more than the process. It has to start with being transformative. That's the number one key. What does transformative mean? Well, it means it's worth working on, right? We have for a long time pressed at, we believe this industry is different. We believe we can change the curve of healthcare. We believe we can change the way healthcare is delivered, sometimes with single doses to effect cures. These are enormous statements, and in our heart of hearts, that's why we're all here, because at some level, we all believe that or some variation of that. And lately, we could see with all of the excitement around the CAR-T space, immunotherapy in general, all of a sudden, a lot of people seem to believe this, and the industry has changed. There's wind in the sails. There's many more people interested now, and people are starting to say, wow, this is really something worth working on. But now we've got transformative therapies. What do we do? Well, we've got to deliver them. We've got to get them to patients. In the journey from bench to bedside, the products are going all the way to the bedside, and they're falling off the edge of the bed. And I think we know that from our experiences watching the pioneering companies that have done great work in trying to make this happen, and we're learning from those experiences. They've been very well effective. They really have been thought through, and they're starting to show us some weaknesses and some strengths in the models that we've put together. So we know from our perspective, people sitting in this room particularly, it's about manufacturing processes, robust manufacturing processes, because the manufacturing processes, obviously, if we can't manufacture in a high quality way, then we can't get the product to patients. So it's a full stop right there. This is a threshold. In many cases, the manufacturing process actually drives the business model, which is really a scary thought when you think about it, right? And that's true for all drugs in some way, but as drugs have become more complicated, it's more and more true. So when the manufacturing process drives the business model, such as short shelf lives, and then sort of really making an imperative as to where these manufacturing facilities need to be, then we know we have a problem, because we've got to be able to control that. We've got to be able to manufacture these at scale with an optimal cost of goods. Now, I put the word optimal here because we don't know, we're never going to have very, very cheap drugs in the regenerative medicine industry. There's too much to do. There's too many hands in the soup. They're very complex products, but we can have better cost of goods. And we can begin to drive those cost of goods through innovation and through engineering and through the right business models. These therapies have to be scalable. They've got to be logistically practical, which means we've got to be able to move them around. When they get to the clinic, they've got to be able to be used by the clinical staff. So one of the perspectives that we have is, having been in the business now for quite some time, we've got well over 6,000 patients that have received products that we've made. So we've got a really good handle on what happens when these products get to the clinical site. And there's a whole series of events that need to be managed, and managed very carefully, in order to make these products the same for every patient, to even make them evaluable, if you will. And then at the end of the day, we got to be able to get them paid for. Without reimbursement, then everything stops. Reimbursement, I like to think of as the sun that pulls the water up through the tree and pulls the whole thing forward. So we have transformative therapies that actually can be delivered in a robust, scalable, and sustainable way. The question really is, are these products transformative? One of the things that got me thinking about this topic is back about a year or so, maybe not quite a year ago, at the Alliance of Regenerative Medicine Investor Day in New York. Olivier Danos was on the podium, and somebody mentioned that the therapies are safe. And he said something that really took me by surprise, because we always say our therapies are safe. And he said, well, they're safe because they're not working. And that was such a challenging thought. I'd never really thought of it that way. They're safe because they're not working. And clearly where he was coming from was these cells that we're used to having cells with a therapeutic index. We push them to the very edge, and that's how we know. So how do we do that in this industry? How do we push them to the very edge of their therapeutic index to know that whether they're not working because they just don't work, or they're not working because we're not giving enough cells, we're not giving them frequently enough? So are our clinical studies designed to yield true positives, or are we just completely missing the boat with many of the therapies? If it works in one patient or two patients, the way these therapies work, you would think that they would work in a vast majority of patients. And yet that we don't seem to see that happening. So before we pat ourselves on the back and say we're there, we really do have to step back and ask, are they truly transformative, and how can our clinical trials really show that these therapies are both safe and effective, and not just that they're safe because they're ineffective? And are we seeing false negatives? So how do we get at this issue of dose? Well, this is something that ever since he said that, it's always bothered me, right? It's always bothered all of us. How many cells should we give the patient? What's the frequency of administration? What's the right route of administration? So we think about these issues and say, well, you know, I know how many cells I'm going to give, particularly in the patient-specific sense, because that's all I can make. So if I can make 1 by 10 to the 9th T cells, that's the patient dose. Well, that cannot be a good way to do this. It's very practical, and I understand, of course, we're practitioners. I understand that if that's all you can make, then that's all you can give. But is that good enough for us? So with dosing schedules and routes of administration, they have been based on some rationale that's come from scientific literature. Again, practicality. If I only have one dose, I can only give one dose. If I can't make more than one. And some preclinical studies. So essentially, at the end of the day, if the question is, are they transformative? And if the transformative nature of these products is actually what makes it worth doing and drives the whole train in the beginning, and we don't know if we're giving enough cells, this is a very serious issue that I think as an industry we need to begin to look at. So the premise, as I mentioned before, is we have challenges. Who hasn't? We're all quite used to this. And going back to that point, yes, we have challenges, but solutions are forming directly along the lines of the unique challenges that are presented. So let's go back and continue a theme that we started in this room a year ago, asking, are we really different? And if we are different, how are we different from previous industries that have actually figured this problem out and turned microgram quantities into gram quantities to be able to make therapies deliverable and in an inexpensive way. So today we're going to look at a few things in this workshop along the lines of the principles of PK/PD, because pharmacokinetics and pharmacodynamics is something we as an industry don't think about too much, because we say we just can't. And I've heard that so many times, it's almost as though this has just become a premise that we all have to live with. We just can't do that. And maybe that's true. And then how do the concepts then apply to us? How do we take those concepts and make them work for us? Because if this is the key to unlock the secrets and to unlock the potential so that we can finally get the therapies into the bedside in a wide scale, if that's really it, then we've got to address the issue. If that's not the answer, we need to move on. But what we would like to happen, the speakers and I would like to happen today, is to get people thinking about this, if you're not thinking about it already. And there is some thought, it's beginning to percolate around that this may be an answer for us, at least something that we should look at. Okay, so without further ado, I would like to introduce our first speaker, who's Dr. Jiwen Zhang, a Senior Regulatory Affairs Director at GE Healthcare, and she's been a leading regulatory strategy development professional for the cell technology and biotechnology. Before joining GE, Dr. Zhang worked in the pharmaceutical industry at companies including Centocor, Johnson & Johnson, and Merck & Company, Wyeth, and Sanofi-Aventis, where she led regulatory strategy development for both marketed products and development compounds in multiple therapeutic areas. And Jiwen's going to give us an overview of what are the principles of PK/PD, because if some people in the audience are anything like me, these are just letters, don't really understand what they mean. And what we'd really like to happen is after we go through the three speakers, we would like to have an interactive workshop with you, particularly those of you who have information about this, and help us start thinking about how this may help unlock the potential of the industry. Thank you. Jiwen.
Thanks, Bob. So let's get to the bottom of it. Let's start with the definition of what's PK/PD. I like this definition. This is from Merck Manual, because I'm a regulatory person, so I like things easy to understand for anybody. So what's PK? Pharmacodynamics. It's really what your body does to the drugs. How it's being absorbed, metabolized, bioavailable, and eventually get out of your body. So if I further simplify it, it's really what level of the drugs after you take will stay in your body, and eventually that level will maintain, and just the clearance out of your body that, to maintain that level to make sure the drug effect can be delivered as intended. So that's PK. PK/PD will always talk together. Why? Because PK talks about body does to the drugs, but PD talks about what drug does to the body. So what does drug to the body is how, when you inject the drugs, how the drugs are going to bind to the target, interact with the target, and eventually deliver the effects. So the level of the drug in your body is directly correlated whether the drug's going to be effective or not. So that's why the drug effect is the PK/PD always go hand in hand. And from PD's aspect, not only about drug effect is going to do what it's supposed to do, but also at a certain level, the drug's going to have off-target interactions, and that's going to cause side effects. So that's why PK/PD are very important, because in my view, it really bridges to the safety and efficacy of the drugs. So this is my very kindergarten attempt to try to draw the picture to see the relationship. But forget about that one. Just listen. Let's look at an example. Advil. So I have a headache from flu, and I take 200 milligrams every 6 hours to ease my headache. From PK/PD analysis, I understand with 200 milligrams every 6 hours, I'm going to have enough level of ibuprofen that's going to last over 24 hours to induce anti-inflammatory effect that ease my pain. So the level of the ibuprofen over 24 hours induced anti-inflammatory effect, that's the PK/PD of Advil. Now think about it. So, I'm tired of taking one pill every 6 hours. Let me just get up and take 4 pills altogether, be done with it. Is that going to work? One scenario is after 24 hours, so I do the PK and I find out, you know what, the level of the ibuprofen is pretty much above the effective level, and it can maintain the 24 hours. So yes, it will control my pain over a 24-hour period after I take 4 pills once a day. Second scenario could be it does maintain the level, but unfortunately, the level will drop to below the effective level after 24 hours. So what's going to happen? That means 4 a.m. in the morning, I'm going to wake up because my headache comes back. So that's not good. Effectiveness is not good enough. Another scenario is the effective level can be maintained over 24 hours if I take all 4 pills at the same time. But unfortunately, because I take them all together simultaneously, the peak level is too high. Now, very likely the chance is going to be higher. I'm going to burn a hole in my stomach. So that's how the PK/PD are very important and useful to really link to the clinical safety and efficacy. So when you're trying to adjust the dose or dose regimen, etc., that's what you're going to start with, then you have a good indicator. The same thing, like my kids, they're not going to take pills. They're going to take liquid formulation. So the same drill. You do the PK and you try to understand the levels of the effectiveness. PK/PD actually is used extensively in small molecule drug development approval, but I think the most prominent usage of PK/PD is really for generic drug development approval, because generic drugs, you don't need clinical studies pretty much. So by statute, the Food, Drug, and the Cosmetic Act requires for generic drug approval, you have to establish pharmaceutically equivalent and bioequivalent. What does it mean? So you go to CVS, you have Advil from Wyeth, now Pfizer. You have CVS ibuprofen. If you look at the label, they're the same, right? So pharmaceutically equivalent, relatively speaking, is not that difficult to establish, meaning you have to have the same ingredient, the same dosage form, regimen, etc., same strength. But normally, oftentimes, the core package to support approval of generic drugs is about to establish bioequivalent. What does it mean, bioequivalent? Again, the FDA requires is about rate and extent of the release of the drugs and action. So it's really transformed from the bioequivalence to pharmacokinetic endpoint. That's what FDA will be looking for to see whether you will have bioequivalence. And essentially, this is translated into 2 key endpoints or parameters. The Cmax, which is the peak level, as I talk about, is really related to, pretty much associated with the safety aspect of the drug. And the AUC is the area under curve over time period. That's associated with the effectiveness, but also for chronic can be associated with the safety aspect as well. So those are the 2 key parameters. And actually, there is a FDA guidance on the PK/PD for generic drugs, and also specific guidance on the PK/PD for specific generic drugs as well. So, hopefully this is helpful to show you, at least in the small molecule world, it's a big deal to do the PK/PD because it bridges to the clinical safety and efficacy. The same framework and principles are also adopted in biologics about pharmaceutical equivalent and a bioequivalent, especially when companies trying to establish biocompatibility. They use extensively PK/PD assessment to support when there are changes being introduced. So my former colleague, actually Honghui Zhou, gave a talk on comparability assessment several years ago, and I want to borrow 2 cases he presented here. Case number 1, injection method change. So if people are familiar with the biologics, there is an evolution of the delivery dosage form, right? From lyophilized to liquid formulation, and then pre-filled syringe, and then now is for sub-Q. Pretty much it have to be auto-injectors. So for this one, just assume it's from patient inject manually change to using auto-injector. So in this case, the phase 3 studies actually was using the manual injection. So patient is injecting themselves. In parallel, the auto-injector was developed and can be in time for marketing. So how to bridge from the manual injection to the auto-injection to make sure the auto-injector can be immediately introduced, in the initial approval. So that's the question. So comparability assessment package. From pharmaceutical side, this case is relatively easier because the liquid formulation is the same. It's really the injection method. But still, the container closure system are different, right? So you still have to do a pharmaceutical assessment, comparability assessment. Luckily, check. So it is comparable. How about from bioequivalent side? So this is where the human PK study is really used to support the comparability. So human PK study is conducted, and predefined biocomparability criteria is with 90% confidence interval, 80% compared new to the old, the ratio has to fall into 80% to 125%. I want to emphasize this one because this is important. This is the conventional or traditional bioequivalence criteria, 90% confidence interval, 80% to 125%. Luckily, they did the PK study, AUC met the criteria, PK missed just a little bit, but still is deemed acceptable. So with this comparability package, the injection method can be switched to auto-injector, and auto-injector can be approved in the initial marketing licensure. Second case, this is a little bit more complicated, cell line change. And I think this can be applicable to the stem cell as well, because cell line A has very low yield. That's kind of like mess up Bob's 2 criteria at least already, right? Not very scalable. It's going to be very costly. It's going to be very costly, so cost of goods is going to be very high. So from the very early on, the sponsor realizes they have to switch to a high yield cell line, from cell line A to cell line B. And if you can manage it, you would like to introduce the high yield cell line as early as possible during development. So in this case, the cell line A products were used up to phase 1. Cell line B products can be available before phase 2. So what will be the comparability data to support switch from cell line A to cell line B? In this case, 2 cell lines could potentially produce totally different products. So the pharmaceutical assessment, in vitro comparability assessment, are much, much more extensive than the first case. So if you look at the FDA guidelines or even the biosimilar guidelines, it's really a battery of the assessment, in vitro assessment, to make sure it has to be comparable. But also, luckily again, in this case, it is shown pharmaceutically comparable. How about biocomparability or bioequivalence? So in this case, they did a monkey PK study. And also, I want to point it out, because this is considered as a highly variable product, they predefine the comparability criteria. Comparing the new to the old is in 90% confidence interval. The range is 70% to 143%, because it's considered variable. So the criteria must looser to give higher probability of success. But unfortunately, with the monkey PK study, the AUC was okay, did fall into the criteria. but Cmax missed. So remember, Cmax, the peak level, oftentimes is associated with the safety. So what are they going to do about to support this process change? So instead of doing the phase 2 study as originally planned, now before treating patients with the new product, the cell line B products, they're going to add a single dose run-in period to a certain number of patients. What does it mean? So I mean, those number of patients are going to receive single dose of cell line B products to assess safety. And if safety can meet the predefined criteria, then they can continue the phase 2 dosing as originally designed. So this is also, in the end, it was a good story. It was a successful case because it was able to support the switch at a phase 2. So unfortunately, I think Bob touched on it as well. In cell therapy space, we don't have the luxury of PK/PD yet. We gave the patients the cells, and we're not exactly sure where the cells go, how long they're going to hang around, and eventually when they're going to get out of the body. That's kind of PK concept, right? What the body does to the cells, does to the drugs. And we don't know exactly how the cells are going to really deliver the treatment effect as intended. That's the PD part. So the question, or one of the solution or proposal here is, can we label the cells and then use the imaging technology to track the cells so we can understand how the drug's going to work and how the drug's going to hang around in the body and eventually get out of the body. So this is very important. These are the very important questions for sponsors to really address for development. But regulators also recognize the importance. So in my appendix slides, I cited, I think, 4 or 5 FDA, EMA, and Health Canada guidance talking about the importance of really trying to, answer those questions. I used the EMA guideline here because not only it talks about it's important, but also EMA was very practical to say, they would like to encourage the sponsors to identify and develop and eventually validate new technologies to be able to address these questions. So that leads to my last slide, and I really want to share with the people here is a new project just has been approved by ILSI, Health and Environmental Sciences Institute. And the project name is Identify and Optimize Novel Technologies to Evaluate Cell Therapy Safety, Mechanism of Action, Efficacy. So I think the sponsors, it's sponsors' responsibility to really identify, develop the technologies to address these questions. But at this stage, it's very challenging for any single sponsor to take on this task. So that's why HESI took the opportunity trying to really pull, the stakeholders together. HESI, if you don't know about it, is a nonprofit organization, and they have established this tri-party platform. So every single project, there have to be participants from the government agencies, academia, and industry stakeholders. And then they can really organize, coordinate interlab studies to address specific regulatory and scientific questions. And they have a pretty good track record to tackle this task. So I put 2 examples here. One is about using transgenic mouse model for carcinogenicity assessment that has been incorporated into the ICH guideline. And then the second one is an ongoing project that actually I'm participating as well. OG is part of it. It's really using stem cell cardiomyocytes in silico modeling to detect, to assess drugs pro-arrhythmia risk instead of doing the traditional hERG assay, and eventually to really eliminate the human thorough QT study that's required by the ICH E14 guidance. So I put the milestones here, but this is really preliminary. The project is going to start next year officially. And if anybody is interested in this project, you can go to the HESI website or let me know. I would like to connect you to the website. But the bottom line is this is a very important area. I hope I was able to convince you how important the PK/PD is. Actually, we were having a roundtable last week at FDA about manufacturing change comparability assessment, and I raised this one. I said, you have cell characterization and you have to do clinical study. You don't have an intermediate like the PK/PD to really bridge or link from cell characterization to the clinical safety efficacy. So I hope this is really helpful to realize it's so important. It's such a valuable to be able to conduct PK/PD, and I think the field needs a little bit more collaboration to tackle this question. Thank you.
Thank you, Zhiwen. That was great. So our next speaker is a chemical engineer who's actually practicing in industry, is practicing these principles and the application of them. Andrew Weber is an investigator in the modeling and translational biology group at GlaxoSmithKline, where he supports pharmacokinetic and pharmacodynamic modeling of macromolecular, small molecular, and antibody-drug conjugate entities. His modeling work has pioneered new methods of physiologically based pharmacokinetic modeling and has been used to support the design of clinical and preclinical trials, including dose selection, therapeutic window calculation, and disease state effects. Andrew.
Morning, everyone. I want to start by saying how great it is to be here talking to a bunch of stem cell industry folks. It's why I got into pharmaceuticals to begin with, the promise of truly regenerative medicine where we can fix these disease states permanently and regeneratively. And my background, as Bob said, is mostly in biopharmaceuticals, and I sit in a group right now that the primary remit is dose selection and clinical trial design. And what we get when we get involved in a project, there's often no human data, maybe even no animal data, and that's a really challenging space to be in. And we want to do our best to say, what dose should you go into first in animal studies with, and then in human studies, and then as the human trials and animal trials progress, we refine those dose calculations. So I want to start with obviously the big problem of the pharmaceutical industry, and the big problem right now is cost. It takes, if you believe or take the Tufts study in 2014 with all of its caveats, $2.6 billion to bring a new drug to market, and 12 to 15 years of research and development and clinical trial time to get there. And what really drives the cost, or one of the factors that's really driving that cost, is the low success rate. In clinical trials, you're looking at between 15 and 30% success rate. And if you go back to R&D, where probably about half the cost of bringing that drug to market accrues, you probably look at a much lower overall success rate of 5 to 10% of all the molecules that come through R&D. So why is it so hard? Why is the success rate so low? And the reasons are it's really difficult to design a drug, design a treatment for a particular disease. You have a lot of things to think about. You have the pharmacokinetics question that we just talked about. Will you get enough drug into the patient? Will that drug stay there long enough? You have pharmacodynamics. What does the drug do when it gets into that patient? You have target selection. Did you choose the right target for your drug? And you have toxicities. I think Juan covered most of those in the last slide. But there is some hope. If you look at reasons for failure going back to 1991, I have the chart up here of reasons, and about a third of them fall into the category of pharmacokinetics. You didn't get enough drug into the patient, or it didn't stay there long enough, and another third fall into the category of pharmacodynamics. So you did get enough drug there, but you chose the wrong target, or you didn't hit that target hard enough. If you look into 2000s, you can see the pharmacokinetics as a function, or the percent of trials that fail from pharmacokinetics shrink precipitously from about a third down to well under 10%. And why is that? For small molecules, primarily, that we're going in these 9 years, And the reason is we got a lot better at molecular design and dose prediction from some of these modeling techniques, particularly physiologically based pharmacokinetic modeling, which I'll touch on later in my talk. So as I said, it's a big problem, and one of the ways that we solve big problems is we divide them into into smaller problems. And I wish I could take credit for this, but this concept comes from a group of pharmacokinetic scientists at Pfizer, and they call it the 3 pillars concept. So what do you need to think about in order to get an efficacious drug? So the first thing is, are you going to get enough drug on board? What's that concentration exposure at the site of action they call pillar 1? The second thing, pillar 2, is target engagement. When that drug reaches the target, does it bind to that target? Does it bind to that target in the microenvironment that target's sitting in? And the last pillar is functional pharmacology. So you have binding to the target, but does that binding to the target actually cause movement of the downstream biomarkers that are causing that disease? And does the movement of those downstream biomarkers then actually cause functional pharmacology that ultimately treats the disease for the patient? So how do we address these? When we're in that preclinical phase, how do we start thinking about, will we have all 3 of those pillars aligned? And one of the primary techniques we use is called physiologically based pharmacokinetic modeling. And the reason we like to use that is because before you know anything about your drug, anything about how your drug's going to interact with the patient, you already know a lot about how that drug's going to distribute, because you know a lot about physiology. You know what the organ volumes are. You know what the flow rates of blood plasma is to those organs. You know how the kidney works, how the liver works. Those you can already build into these models before you know anything about your drug. And then as you get more and more data on your drug, you can refine these models and make better and better predictions as to how much drug you need to get on board in these patients, or how much drug you will get on board in these patients at your given dose, and be able to rationally do dose selection. So with these models, as you progress through the clinical phases, getting good data to validate these models and to calibrate these models is key. And just wanted to show a quick example of one type of data that we collect to calibrate our models. And in this case, what we did was we took a domain antibody, so it's a 25 kilodalton protein that we want to use as a platform for a lot of our different biopharmaceutical drugs. And we put a radiolabel on that, and we injected it into a mouse model. And then you take that mouse model, and at different time points, you can then section the mouse, and you can actually examine where that drug distributed in the mouse, and look at where your model's predicting relative to that distribution. And on the lower right corner, I'm just showing the blood curve and a curve in one of the tissues, the kidney, and the dots are the data, and the curves are the model predictions. And you can see it fits the data quite well, and we have 13 other organs that we looked at, and it fits the data quite well in these models across all the organs. So we have pretty high confidence that we know how our drug is going to distribute within the body, and that it will reach the target or won't reach the target from these models based on the validation. So what does this mean for cell therapy, and how do we go from biotherapeutics to cell therapy? And I think the actual fundamentals of the models are the same, because the physiology is the same. You still have a person, they have the same organs, the same blood flows, but it's all about getting the distribution mechanisms right. So what's going to carry those cells to the different tissues and different spaces you might want those cells to have their functional pharmacology. So there's some similarities between the biopharmaceutics and cell therapy areas. So with both of the molecules, they're confined to the extracellular aqueous phase. They're not going to easily cross plasma membranes. But there's a bunch of key differences. So for biotherapeutics, when you think of extravasation, so what's going to drive those biotherapeutics out of the blood system and into the tissues where you often want to have your pharmaceutical effect. So for biotherapeutics, you're looking at a mechanism of filtration. So those biotherapeutics are driven through pores in the endothelial walls, and the mechanism that's driving them there is that you have a pressure difference. You have blood pressure, and then there's much lower pressure in the tissue spaces, and that's going to drive some flow across there. That's going to carry the biotherapeutic. For cells, you'll probably have that to some extent, But what I suspect is probably more important for cells is understanding the chemotaxis that drive cells to actively try and transport themselves into one tissue space or another, actively leave the bloodstream. We also consider in these models effective dose for biotherapeutics. And for biotherapeutics, for small molecule therapeutics, it's always less than the delivered dose. You'll never get more drug in there than you administered. But for cells, this is not necessarily the case. You have cell division, you have cell proliferation. So your likely effective dose will exceed the delivered dose. And I think one further thing to think about this is the differentiation. If you're putting multipotent stem cells in there, how many of those stem cells will actually divide into the efficacious cell type that you want to cause your pharmacology? You can look at elimination, so what's going to drive these drugs to leave the body. And for biotherapeutics, you're looking at degradation by proteases, and for smaller biotherapeutics, glomerular filtration, the kidney removes a lot of the smaller molecules. For cells as well, you have some mechanisms that might drive them out of the body or drive them to no longer be efficacious, things like apoptosis, cell death, cell killing, maybe things like immunogenicity, which are probably applicable both for cell therapy and biotherapeutics. Does the immune system recognize these things as a foreign invader and will try to carry them out of systemic circulation? And lastly, within each of the tissues, so for biotherapeutics, within each of the tissues, once you get out of the vascular space, the diffusion of those molecules is actually really fast in there. They're really well distributed in the microenvironment of the tissue. But for cells, that might not necessarily hold. Maybe the chemotaxis in there will drive them to distribute in some areas of a given tissue, but not others. So by no means a comprehensive list of things to think about, but certainly at least a good list to get started is to thinking about what might be the mechanisms if we want to derive these kinds of models for cell therapy. So what about pillars 2 and 3, the actual efficacious side? And that was all great for pharmacokinetics. And I think the tools we're already using for small molecules and biotherapeutics will actually be quite useful for cell therapy as well. And the reason they're quite useful for across these platforms is once you get target engagement, all you really need to worry about is what the pharmacology is that's going on in the patient. So the downstream effects will be just governed by the patient biology and not necessarily by the treatment itself. So when we look at measuring these or look at modeling these and predicting these, it broadly falls into two categories. So you have empirical models, so that's you take your drug of interest and you put it, or in your case, your cells of interest, you might put it in a petri dish and incubate it with the target, and you measure the effect over time. So if you're doing something like an antibiotic, you might measure how many of those cells are killed over time at a given drug concentration. And then you can fit some rather simple mathematical functions, something like a Hill equation or a logistic function, to theirs, and then predict based on your drug concentration over time what your effect is over time pretty simply. But more recently, if you remember back to my second slide, pharmacodynamics are still a big driver in why a lot of these molecules fail in the clinic. More recently, you have more advanced modeling techniques that actually look at what are the mechanisms that drive to this functional pharmacology. How does when your drug engages a target, how does it actually affect all of the cell signaling in something like systems biology? And then if you go in and go up one level, how does that cell signaling then affect tissue signaling and the eventual functional pharmacology in a modeling construct known as quantitative systems pharmacology that's really just starting to be used more widely in the industry. And the interesting thing about quantitative systems pharmacology and modeling all this signaling is the toxicology end of things. So if you remember that second slide, toxicology was also about now 25% of the reasons for failure in the clinic. And pharmacology and toxicology are actually rather similar if you look at them from a mechanistic standpoint in the body. So you can actually apply these quantitative systems pharmacology to a field called quantitative systems toxicology and start predicting some of the target or drug toxicities you might get in the body as well. So where does this leave us? So what's the path forward with this? So I think that the first thing we would want to do for cell therapy is get some idea of the time course of cell distribution around the body for different types of cells, what's driving them into the tissues, where certain types of stem cells going to land in the body, and we can then use these to develop platform models we can use for wider predictions, and maybe it'll probably be somewhat expensive at the beginning as you need lots of imaging data, but as you establish these platforms, they get acceptance, that then you can do less and less of the imaging and rely more and more on the mechanistic data you already have. We want to derive some rules for what's going to drive the cells out of the plasma space and into the blood vessels, and I think there's already a lot of good data available in the literature you can use to look at deriving those rules or look at calibrating those rules against. Describing cell-cell interactions is a quite interesting field, so both in terms of the division of cells, creating, as I said, that additional bioavailability of your cells in the body, but also in the actual target engagement. So what do you need to design your cell? How many receptors does that cell need to express to engage another cell in the body, the target cell in the body? And then also describing the downstream pharmacology through methods either like the empirical modeling or the quantitative systems pharmacology, and then coupling that to the pharmacokinetic cell models that we previously developed. So thanks for your attention, and hopefully generated some interesting discussion points for our upcoming forum.
Thank you, Andrew.
Thanks.
Okay, Andrew, thank you. That was great. So we've gone from a broad regulatory perspective that Jiwen provided for us and into a bit more the science of PKPD. And now we're going to slightly change tacks a little bit and come out of the from the cell therapy industry perspective with a very interesting perspective, an interesting person. Frank is a physician scientist trained in clinical pathology and immunology, and he's got a broad practical hands-on experience in all aspects of business development as well, including search, technical due diligence, commercial, financial, and industry sort of strategic evaluation experience. He's been a pharmaceutical industry professional since 1999, beginning in clinical research and transitioning to business development by way of a 7-year stint in the financial industry as a biotech buy-side analyst. And so now Frank is a BD professional since 2008 with Millennium, started with Millennium, which became Millennium Takeda Oncology Company, went through Shire, now head of search and evaluation at Baxalta. So I think this is going to be a very interesting sort of turn, I think, and I want to put one more point out before Frank gives his talk. As we talked about those hurdles and the challenges as we go from bench to bedside, if you take a 30,000-foot view and look at the industry in terms of starting out with manufacturing processes, which we mentioned before, the business models that they drive, and then finally, what does this need to look like so that we have robust, high-quality manufacturing at an optimal cost of goods, scalable to meet the demands in a sustainable way over the commercial life of the product. We've got a story that we have to tell that says today's manufacturing processes are not that great, by and large, and in fact, we don't know any that we would say, boy, that's a great manufacturing process. We've got to figure out better business models as we're trying to fix those processes and then look down the road to what we like to think of as the factory of the future. At Baxalta, they've been working on this for a number of years under different types of names and have been leaders in the industry and really thinking through how do we change this paradigm, because if what we have are clean rooms and what we have are the processes we have today, then we don't have an industry, period. And I would say way back to Baxter immunotherapy days, all the way up through Baxter, Fenwal, et cetera, all the way through to Baxalta now, they've been thinking through these problems. So we'll ask Frank to come up and give us a perspective from that side, and then we can all have a conversation. Okay, thank you. Dr. Borriello.
Thank you very much.
You're welcome.
It's a pleasure to be here to speak to you today. Many of you may not even know Baxalta. Baxalta is a recent spin-out of Baxter Pharmaceuticals. It went live as a public company a couple of months ago. But because of our pedigree, we come to the market with significant revenues in product sales. And we also come with significant experience in both cellular therapy as well as biotherapeutics, both in immunology and hematology. I want to thank my previous two speakers because I think they set it up very nicely. What I think they've said in a nutshell is that cellular therapy is not immune to, or exempt, I should say, from the basic principles of drug development. And I think as much as we try to say that cellular therapy is something special and should have its own rules, the bottom line is that it enters a certain regulatory paradigm and a certain way of developing drugs in the Western world that we have to adapt to. Now, PK/PD for small molecules is hard enough, but PK/PD for cellular elements is excruciatingly difficult because of the issues involved. Cells are really complicated. I mean, there's a lot going on there. I dare say that a lot of the cells that many of us work with are not fully characterized. We actually don't know what the secret sauce is that makes them do what we think they do. They are not easily measurable. Once you put them into a patient, you can't necessarily just take a blood sample and say, wow, how are they doing today or 1 week from now? Oftentimes, they extravasate out of the vascular space. They take up residence. They may live for variable periods of time, sort of induce an effect and then die, as is the case for, I think, most mesenchymal stem cells. It's really hard to measure. Pharmacodynamics, however, give you another leg up because what you're looking at in pharmacodynamics is the downstream consequences of having given your drug. Sometimes the choice of indication you go after gives you easier or more difficult pharmacodynamic options. I think just to pick an example which will lead me into the 2 or 3 slides I want to show you is some of the cellular therapy work being done in the hemoglobinopathies. So for sickle cell disease or beta thalassemia, it's quite clear what the genetic defect is. It's a defect in the beta globin gene. It's easily visualized what's going on in the periphery. And similarly, if your drug is having any effect, if your cellular drug is having any effect, it should be manifest in the peripheral blood. So you clearly have a pharmacodynamic marker to follow. And I think it's not by accident that some of the most advanced cellular therapy companies have focused in areas where pharmacodynamics are more readily measurable. I mean, we're trying to collect the low-lying fruit here before we get to really complicated stuff like going into the CNS where sampling is impossible and you're looking for long-term consequences that can only be manifest over years. Think about that for drug development. That would be a nightmare. And so there's a reason why companies have started in the hemoglobinopathies. I want to get to another issue, and that is characterization of the material we put into patients. This is CMC 101. Whenever you're talking about a small molecule, you have to characterize it down to the atom. You have to be able to demonstrate that you can create and characterize this molecule at all its intermediate stages. You have to show that you can do it reproducibly every time, that there's tight variances in the drug that you create. Now, shift to cellular therapies. If you're isolating cells from, adherent cells from bone marrow, well, do we really have one population? Do we have a mixture of populations? Is it going to be the same from time to time that you do it? These are questions that are being addressed by people that are a lot smarter than I am, but they're difficult problems. And I think it would truly be a shame if some of the clinical trials that are ongoing fail because the material put into the patients was not consistently characterized and reproduced. That would be an utter shame. So thinking about those principles, we developed a device at Baxter, which Baxalta has now inherited. Next slide, please. So I have the... This is a device that we're planning to use for some of our studies in cellular therapy. And we're interested in both the immuno-oncology space as well as the hemoglobinopathy space. And what it is, it looks very simple. It's a standalone device. It was originally designed as a point-of-care device, although depending on what applications you have in mind, it could also be a back-office device where you use it just in a manufacturing facility and provide the product. But the reason it kind of looks nice and sleek is because you can imagine this being right next to someone's bedside. And it builds on a previous device that we had. This uses magnetic beads or ferrofluids. We're developing new versions of magnetic separation technologies for both for positive and negative selection. And that of course implies you have markers that you can positively and negatively select for, and that you have antibody or nanobody or aptamer tools which allow you to tease apart specific subsets of cells from complex mixtures. So that already speaks to a level of characterization that you have to have about your cellular therapy that I don't think everyone currently can claim. This system was built with the idea of the regulatory bodies in mind. Because like I said earlier, you have to prove to the regulatory bodies that you've characterized what you're putting into people. It's a safety issue. They're not just going to let you put any ill-characterized mixture of cells into people. The idea is that it's fully customizable, automatable, and regulatory compliant. So the software, which obviously you cannot see, is extremely modular. You can imagine that as you're working on this device in the laboratory, you can record all your steps. You can automate it. It'll be done the same exact way every time. We're at the point right now where the key element here is a spinning vessel, which I'll show you a picture of in a moment, that doesn't require any centrifugation steps. So think about it. When you centrifuge cells, what do? You're bunching them together in a rather unnatural way. You're decanting the supernatant, and then what? You're banging at the bottom of the tube to sort of resuspend them. What does that do to the cells? What if the cells are sensitive to cell-cell contact? What if they're sensitive to differentiation, as many of the hematopoietic cells are wont to do? So this is sort of the heart of the machine. It's a spinning membrane which allows you to wash and concentrate the cells that you've previously separated with your magnetic beads, and it allows you to do it the same way each time. There's no bunching up of cells. It's all in a closed loop system, so sterility is guaranteed. This is a schematic, and all these bags and tubes, which look like spaghetti, is actually a disposable kit. So you put this disposable kit on. It has various reservoirs that you can customize. You have your wash solutions. You have growth media that you can apply. The little rectangles are computer-driven valves that you program, so all the washing is done the same exact way every time. And of course, then you end up with a final product in one of these bags, and you're able to administer, well, first of all, you characterize that again before administering it to the patient or freezing it down for later administration. So this is the kind of tactical steps that I wanted to show you that Baxalta is taking to reduce to practice some of the things we talk about generally. I mean, the problem of cellular therapy is huge, and if you look at it as one monolithic thing, it can be a little overwhelming. But we think we're addressing a particular portion of it, which quite honestly, in our talks with collaborators, they never think about. CMC, we like to say, is one of those things that unless it's going terribly wrong, you want to ignore it. And this is going to help our collaborators understand that they can have a reproducible system at their service so that they can worry more about the front end, characterizing the cells with their whatever monoclonal antibody reagents they need to develop, and they can characterize the cells in other ways. But the downstream manufacturing front, I think we are doing a good job at getting down in a systematic way. So those are my formal comments. I hope that we'll be able to have a discussion later on. Okay, if there are any questions, please.
Just have a seat there. Okay, so thank you. That was great. So I think as I mentioned, we've gone all the way through from regulatory through science and through some practical application. I guess I can, before I start with questions and comments, and the panel just starts to speak, why don't we open it up to you? You've heard a lot this morning. Is there anything burning on people's minds you'd like to challenge the speakers with? Do you have any perspectives to add? Yes, sir, please. There's a microphone coming. Please introduce yourself.
So bottom line, I guess for the PKPD and cell-based therapies, is cell fate going to be kind of the set that describes what's ultimately going to get it through regulatory approval, whether it's the therapeutic fate or some kind of, you have to invoke cell death, anything on that order?
Could you define what you mean by cell fate so we're all on the same page?
Yeah, well, so if you have like a regenerative therapy or some kind of restorative therapy, like what happens to the cells, or versus, say, a delivery kind of therapy, do the cells die? Do they become incorporated? Do they have to be around forever? Is there a time course? And, program cell death.
Okay, well, it's a great question. Actually, I wrote something down during Andrew's talk because he talked about extravasation, driving force, effective dose, target engagement, elimination, and then compartments and circulation. And I think your question is to that elimination point, but you also mentioned something about integration. And you didn't talk about integration at all. You talked about eliminating apoptosis, cell death, the like. But in the issue of cell fate, one of the difficulties is considering, even if you select the cells out using Bexalta's device and you've got CD34 pure cells, they get there, how long do they last? And how does that completely throw a wrench into everything that you like to think of very neatly in your world?
Yeah, I think it's definitely an additional consideration there. I think when you think about these, at least when I think about these therapies, I think about two things. You have the kinetics of what's going on. So you have that initial sort of distribution phase, the cells are working out their way, and then you probably reach some sort of long-term steady state there. That's sort of what I interpret as your eventual cell fate. And what matters, at least to me, I think, is probably dependent somewhat on the disease that you're trying to treat. Is it something that you can get away with just having a high spike in the beginning and that resets the body into whatever its pre-disease state mode was? Or is it something that you're going to need chronic exposure to cells for? And then obviously working in those mechanisms, understanding those mechanisms of integration and long-term cell fate become critical. On the regulatory end, I'm not sure that I have a great answer for that at this point. I would imagine the regulatory probably somewhat will follow the science as that gets published and people understand more and more about these therapies.
So GUN, will it all come, I think the question is, will it all come down to cell fate? I mean, is that, because I kept thinking the same thing too. What's the stake in the ground here around which we start to, so is it about cell fate?
No, it's not, right? If you think about it, some of the drugs got approved without understanding of how it works. So there are precedents that drugs can be approved without, all this human PKPD understanding, as long as you can show they are safe and effective. What PKPD offers you is you have a valuable tool. So from the product to the safety and efficacy, you have something in between that will help you to establish the safety and efficacy. So to answer your question, if you don't have to do it, you do the clinical trials and you show the safety effect, you're going to get approved. That's why FDA or any agencies, in the guidance, they talk about it. It's important to do all of this, but they're not enforcing it yet because they know right now there are no technologies that really help you to answer that question. Does that answer your question?
Yeah, it does. It kind of goes to I guess imaging is going to kind of be incorporated, more and more, just to sort of see where they are and what they're doing versus kind of like, do they disappear, which usually seems to be what happens in a large number of cases. But I was mostly thinking in terms of it looks at this point like therapeutic efficacy is the end point, and it's kind of a however you get there through the PKPD is going to have to be acceptable until you can raise the criteria for definitions. I mean, obviously the old bugaboo with stem cells was, they're going to cause cancer. So that's going to be kind of a therapeutic or lack of therapeutic endpoint.
So if you can show using the imaging or whatever technology to show, you know what, stem cells you inject, they're not going to the target, they're not going off the target, and even on the target, they're not going to really further differentiate into something you don't want, then you're good, right? Then you don't really have to use hard clinical endpoints to address those questions, meaning you have to collect patient data to say, you know what, I'm not going to get tumor from those patients, which is very difficult. So at the end of the day, PKPD are valuable tools. The hard endpoints are still clinical endpoints. So this is really the bridging.
Yes,
I have a question for Dr. Weber. Can you really neglect sort of the interactions of a biologic with plasmalemma and extracellular matrix as a compartment that it potentially affects the pharmacokinetics?
Who was that for?
That's for Dr. Weber.
Dr. Weber.
Sorry. Can you repeat the question?
Can you neglect plasmalemma
hoping that wasn't for him.
and non-specific interactions with extracellular matrix as a pharmacokinetic compartment?
Okay. So there are interactions
systems.
between say a monoclonal antibody and things in the plasma. So I think it's definitely been a point of contention in the biopharmaceutical industries, and I think there are assays around that look at bound versus unbound drugs. If you look at some of the ELISA, there's some sandwich cultures that are anti-idiotypic that you can detect both bound and unbound drug to things that are in the plasma, and then you have the ones that are non-anti-idiotypic that will detect only unbound drug to things in the plasma. In terms of what especially biologics are going to bind to in the plasma, you get very, very specific affinity, very, very specific selectivity for your targets. So in terms of protein binding, it's fairly negligible for non-specific, but in terms of your target, if you're hitting a plasma-bound target, then you have assay techniques to deal with that.
Frank?
I wanted to add that, first of all, the variety of cellular therapies that are represented in this room are so broad that you probably do need to have conversations with the regulators that are specific for your program. So interaction with the regulatory agency is key. There is no one size fits all. But I also wanted to bring up an example that involves gene editing and the ultimate fate of cells, which I think is what your original question was maybe addressing. Gene therapy, as you know, is a fascinating tool. There are many approaches to doing this, and yet the risk-reward of doing that varies from indication to indication. So if you're worried about leukemias forming because of off-target gene editing effects, and some of these patients are young, so they can live 20, 30, 50 years. The safety hurdle that the agencies are going to put on you is going to be much higher than another approach, maybe in the CAR T space, where, especially in the allogeneic CAR T space, where the cells are expected to die within 4 to 8 weeks. So you can afford to be a little bit wrong or off target in your gene editing. So I always keep those two examples in my mind because it's always a risk-reward ratio that will dictate whether or not you can use a certain technique in a certain setting.
Linda Marban from Capricorn. I just would like to also address the issue of cell fate. I think that we have to be very careful in recognizing that we talk now about cell therapy, and we've moved a lot away from stem cells and integration into the tissue and becoming part of whatever functional organ we're addressing. We have certain cell types, and I think Dr. Borriello just said it perfectly. An allogeneic cell, for instance, can go in and function as a colloquial local drug delivery system where they release their happy factors, they do what they're going to do, they stimulate tissue repair, and then they go away. And from a regulatory standpoint, somebody else just said, there's also a much lower bar for safety because you don't have to worry about a cell embedding itself somewhere else and becoming a cell that you wouldn't want necessarily in your pancreas or your brain if you're putting, for instance, in a cardiac cell. So I think we have to be very careful and not generalize cell fate because there's very different levels of therapeutics that are developing across this wide array of cell therapies that are developing. So thank you for your time today.
So Linda, with with the cell that you expect to integrate, then if you're really expecting it to integrate and do its action over a long period of time, how do you control that, so to speak, right? I mean, and maybe I shouldn't say how do you control it, but in the measurement phase now that we're in the development phase in the industry, how do you predict what will happen in that setting and get a comfort level around that?
Yeah, I like to take a pass on that. I'm a strong believer that our therapeutic paradigm is not advanced enough to really deal with the cell that goes and embeds and grafts and becomes part of, at least in the heart, the functioning syncytium. So, I think we're very lucky that our cells don't stick around because I think the regulatory barriers are going to be very high there. But perhaps some of our colleagues that are taking on this enormous challenge can better comment on that than I can.
I would say that, Bob, you've raised the most difficult scenario, where
the moderator's prerogative.
A cell is expected to be operating in a host for decades. That is a really difficult safety issue to address. You can measure pharmacodynamics all along the way and make sure that the cell continues to provide the effect that you're looking for. You can do that. But I think the scenario you raised probably will only be applicable in some of the most severe diseases right now and not be used where alternatives, short-term alternatives or classical therapeutics are available.
I think as I spoke with Sven Keely from GSK about this very issue, and they're working very hard on this issue with cells. And his question is, what's the dose? We talk about dose. In a situation in which you have stable engraftment of the cells, we can't even tell what the dose is. And so, as you keep going back to what's the stake in the ground that we have to worry about? Is it fate? Is it dose? Where is that? It is a difficult challenge, huh? Well,
I believe Andrew mentioned that in the instance where the cells are expected to divide, dose ranging studies are particularly inappropriate because 1 million cells given at day 0 is 1.5 million cells in 12 hours, and so on exponentially until they some cells divide that fast.
What are the magics of
characterizing the division rate and eventual steady state, number of divisions?
So basically, it's a big black box. You give the cells, you hope for the best. You don't know what exactly happened.
Although I do want to challenge that paradigm a little bit, because if you look at the biologics industry, probably back 70s, 80s, it was primarily the blood products industry, and what they were doing essentially is cone fractionation, so ethanol separation of whole blood into its constituent parts, and administering things like immunoglobulins to immunocompromised patients, factor IX to people who lack the clotting factors, and there it really was that kind of paradigm, right? You administered these very uncharacterized masses of this certain fraction that came off of when you crystallized with ethanol at a certain temperature to a patient, and it had an effect. But fast forward 20 or 30 years, and we definitely can't get away with anything like that. We did what to patients at that point?
I have to take exception with that a little bit because people today are still getting plasma-derived products, and in fact, Baxalta sells some of them.
They are. But would you be able to get them approved in today's regulatory?
That's
really difficult.
But that's my point.
That's
right?
absolutely right.
If you can, establish it's safe, effective, use whatever methodology you can use, that's fine. You will get approved. Neuropathic pain, nobody knows how it works, but somehow it got approved, right? But if you understand this big black box, what's happening to the cells, eventually the fate or the dose or the division, etc., that will really help you to address some of the safety efficacy questions indirectly, because those are the indicators.
But we heard before, so the whole industry started because we turned over mechanism of action to cells. We said past therapies have been great. They've moved the bar, but they don't work in the long term. Cells know what to do. We don't know what they do. So we're going to throw them in there and let them do the work, right? And then at the same time, we want to make switches to different instruments. We want to grow the manufacturing processes. We want to do better PKPD. We want to understand the mechanism. But we've sort of abdicated that to the cell. Now we get into a day and age where we can actually genetically modify these cells, and we can start to turn switches on and off, which finally gives us something we can hold on to, right? We think, at least, that we know what they're doing, and now we're saying they're too dangerous, right? When you do that, that might be too dangerous a thing to do. But I think is mechanism of action, not having that handle on mechanism of action, is that such a serious problem that we can't use these principles? Maybe I'll start. Yeah, I mean, is it a regulatory challenge or is it a business challenge for us? Right?
Regulatory, actually, this is a very good contentious point, because last week at the roundtable, they were saying, ultimately, it's really a scientific question, it's a business question. But unfortunately, business always look to the regulators to say, what exactly do you want? So I can help myself to make that big business decision.
Yeah, but the regulators don't insist that we know the mechanism of action.
No, they don't.
Right, they don't. So, because they know we don't know, right? And so then we want to make a change in the manufacturing process. We want to scale, we want to be sustainable, we want to lower the cost of goods, but we can't. So it seems to me that understanding the drug is really more of a business challenge. The regulators understand the challenge and say, okay, do it. If it doesn't seem dangerous, go ahead and do it. We'll help you watch what happens. And somebody said it before, it's what happens at the end. We give the cells and what happens at the end.
But if you look at what I talked about it from a generic drug point of view and from biologic comparability point of view, once you know that black box, you can convince the regulators eventually. You use this understanding of the black box to mitigate some of the clinical safety efficacy investigation. Right? That's where eventually it's going to be really valuable. And that already has precedence with the generic drugs and with biologics to show the comparability.
But generic drugs and biologics are definable, right? I mean, do we all have to go down the path of understanding and using an Isolex or something? Right?
Do we have to, right?
Yeah, do we have to get that far?
I think I think we're mixing two things, though. What the regulators require is one thing. Yep. They require safety and efficacy. You do that, and you get an approved drug, no matter whether it's cellular, small molecule, whatever. You don't have to know the mechanism, but there's a big but here. As drug developers, if we don't understand what's in the black box, then we are shooting arrows at a target in the dark. You either hit it head on and you say, we got a drug, or you have no way of knowing if you got close and you should just simply adjust a little bit. And we don't have the resources to just take shots in the dark. So the better you understand your drug or your cells and how they operate, the better off you'll be. So that way, when you get an adverse event in the first patient, you'll know how to adjust. Otherwise, you go on clinical hold and you're done. So as drug developers, we should want to know how things work. The regulators are focused on safety and efficacy.
Any comments? Oh, Stuart, I was going to call on you if you didn't raise your hand. So.
Oh boy. Okay. So I'll put in a blatant plug to begin with. So I work for Fate Therapeutics. So Linda, yes, it is all about fate and function of cells. But that aside, so maybe for Andrew, but for the rest of the team. So arguably, one of the single biggest advances for small molecule pharmacology was Chris Lipinski's rules on absorption distribution, so structure-activity relationships for small molecules. We, as a field, we currently have something like 34 ongoing clinical trials for CD19 CAR T cells, just CD19s. All of those, many of those different trials have different antibodies targeting, they have different vector systems, they have subtly different cells, they have different signaling domains. So how close are we as a field to getting to structure-activity relationships, basic structure-activity relationships for cell therapies, given that we have a lot of interesting clinical trials going on that can perhaps give us clues into some of the key elements of that might underpin those structure-activity relationships?
Yeah, I think certainly once you get past the first round of clinical trials now, you have a lot of data to play with and a lot of sort of different variations on a theme to look at what worked better than this or that. You have to, when you go back and want to interpret these trials, you have to go back and consider the whole fate of the cell. Obviously, with respect to that, was it the actual sort of processes that are distributing the cell, or was it the mechanism, the design of the receptors on the cell that caused the difference in efficacy or difference in safety profile? But in terms of understanding these things, at least I think we're taking maybe some of the first baby steps in there. I've seen recently in the last 2 years the first couple of models of cell-cell, three-dimensional cell-cell interaction to try and give you an idea of how you actually have to design your receptors and design the affinity of those receptors in a similar way that we think about defining the affinity of a drug. Now, in terms of the actual distribution properties, I think we're a little bit farther off other than for some of the cells, like T cells, we have quite a bit more data. Just needs to be integrated into a modeling platform. And another interesting point, I think, that you look at, if you look at the history of small molecules and going in this mechanistic understanding, people have been playing with these mechanistic understandings, I think as early as 1937 is the first paper you can trace it back to. But before you started to get regulatory buy-in and true industry buy-in, it took maybe about 1997. You had two companies come out about the same time with Simulations Plus with GastroPlus and SimSip with their eponymous product. And what that really did to the industry is it industrialized these techniques. Now everybody's using the same methodologies to examine these molecular properties and these distribution mechanisms, and then the regulators really understand these and also can really buy in, and that's where you get your big payoffs on the back end, because now you can do things like pare down clinical trials with the mechanistic understanding you have of the drug. You can go into special populations, sometimes without clinical trials, with the mechanistic understanding. The regulators are buying into that, and they're now, I think as of 2012, requiring this for all pediatric clinical trials. The FDA came out with a position paper and said they require physiologically based modeling for all pediatric trials and strongly encourage it for all other trials.
Good. Thank you, Andrew.
Actually, I was going to ask you another question about mechanistic modeling, because that's the future, right? But like even for the CIPA thing, for pro-arrhythmia risk assessment, is really the premises is we think we understand how the drug interacted with the ion channels, and eventually it's going to lead the pro-arrhythmia.
You can build a model around that.
Right.
Yep.
So, but I think the problem here is, in order to build that mechanistic model, you have to understand the things, right? And then you can model it. My question or my kind of worry is, how much data eventually is really, how you define the data is adequate so you can build that model then that can be used.
Oh, that's actually a really good point. So I'll answer the question with my own question, then maybe do some of the thoughts on that. But do you need any data to build a model of a system? So I'll pause for a second, let you think about that. But I would say, no, you don't need any data. What you need is some idea of what the mechanisms underlying that system are. I mean, you might not have a lot of confidence in that model, but building that model forces you to sit down and think about, these are all the mechanisms I need to go through in my system. And then when you experiment, you've already engaged. I mean, you have assumptions, but you have assumptions when you're just thinking about a problem and don't have a mechanistic model in place, and then it actually forces you to say, this is my model, this is what I got in the data. What did I get wrong in my previous model when the data got out, or what did I get right? Is it just that I'm off by how fast these things are happening, or is there something else, a completely different mechanism that I'm missing? And that can be a great sort of Bayesian reasoning tool to help guide you through thinking about mechanistically.
Don't let perfect be the enemy of good.
That's a very good point.
And get started. Interesting. Frank, did you have a comment?
Yeah, I just wanted to say that the CAR T cell space is really intriguing, because I think they're doing it absolutely right, and there's a reason why they have certain advantages there. Clearly, the risk-reward profile when you're talking about cancer patients allows you to do things that you couldn't do in an otherwise healthy population that's going to live for decades. Also, the fact that there are 34 permutations of the study being done in CD19 alone is great, because it also tells you, I don't know if everyone's familiar with the data so far, but in the first 10 patients, you see an 80% response rate. So it also, with a very small number of patients, which equals low cost, you can see if your construct, if your variable has improved or worsened the situation. It lets them do exactly what I was talking about earlier, of understanding their variables, how tweaking it in one direction or another is manifested in efficacy or safety. And that's, I think that's why the CAR T cell space is probably making cellular therapy a reality. Some of, well, what Bob brought up earlier about something that's expected to live inside of you for the rest of your life is a problem that's another order of magnitude more difficult. But the lessons learned from the CAR T synthetic biology space is exactly what is going to help the rest of us do things with mesenchymal stem cells, for example.
Can I just one
more
last
point?
point?
Yeah,
Yes.
good.
I think we talk about CAR T and everything, and people also trying to anticipate eventually it will be personalized medicine. On one side, I'm saying, as long as you can show efficacy and safety, you're good. But if you really want to eventually to achieve personalized medicine, you have to understand PK/PD. Otherwise, you cannot really personalize. Because safety efficacy is a population data. It's not individual data. So, that's PK/PD eventually can really help you to really achieve it can be personalized.
Very good. Thank you very much. Thank you, panelists. And I'll have one comment, is go forth and multiply now. Have a great day. Enjoy the rest of the conference. Thank you.